Okay, in this unit, we're going to be focusing on the cell cycle, what it is, the different parts, but it's going to be the cell cycle for somatic cells only. So let's do a quick review before we start looking at this stuff. There are different types of cells in the body, but there are two main categories. You have somatic cells, and then you have your reproductive cells. Your somatic cells are just everyday structural cells, like skin cells and heart cells and eye cells and brain cells and liver cells. So it's all of the cells that make up the main parts of the organism. Okay, those are somatic cells. Reproductive cells are something different. They are not considered somatic cells. So what we're looking at in this particular unit is just cell division for somatic cells, whether it be a plant, animal, insect, fish, whatever you're looking at. This cell cycle unit is just for the somatic cells of that organism, not for the reproductive cells. Okay, so why would somatic cells need to divide? Why would, say, the cells that make up the stem of a plant need to divide? Or why would the cells of our skin need to divide? Well, obviously growth is a factor. Okay, organisms grow, and as they grow, those cells are going to need to divide. There's going to be times when cells will die because they get worn out or they get damaged for whatever reason, so they need to be replaced. Cells need to be specialized, right? If you look at a plant, there's leaf cells and stem cells and cells that make up stuff that allow things to flow, and flower cells and plant and fruit cells and all different types of cells. The same thing for animals. You have skin cells and the different cells that make up the organs. So cells need to be specialized, and so cell division would occur because of that. And there are limitations due to size of a cell that would make a cell need to divide. And that's also important because once a cell gets to a certain size, it runs into issues. And so let's look at some of those real quick. Cells, obviously, for eukaryotes are different sizes and shapes depending on what the cell is used for, what the cell specializes for. Okay. However, it can't get too large. The reason being you don't want it to get too large is because it can affect things like the amount of energy needed to maintain that cell. It can affect things like cell transport once the cell gets too big. Think about it, as a cell gets bigger, the outer membrane, the cell membrane, is actually getting bigger too. It's increasing surface area. When you do that, it's going to require more energy to make cell transport work, for example. Okay, it's going to require more energy to maintain the large size of that cell. So those are all issues that how size can limit a cell's functionality. If you take this picture, for example, as the cell size increases, its volume also increases, and volume means the space inside of it. Okay. Also, the area around it is going to increase. All right. When the cell size doubles, for example, it's going to require 8 times more nutrients, 8 times more waste to get rid of. It's going to need more energy to use those nutrients and to get rid of that waste. The surface area is only going to increase by 4 times as much, but still, the more surface area, the decrease in the ability to transport. Okay, so diffusion and osmosis are going to decrease. So those are some limiting factors to cell size. Okay, a reason why you don't want a cell to get too big. Now, obviously, we have 2 different types of organisms. We have eukaryotes and prokaryotes. Think about the big difference, and the big difference is your eukaryotes have organelles. Okay, so cell division for eukaryotes is more complicated than cell division of prokaryotes, or your bacteria. So we're going to be looking at first the cell division for eukaryotes. Okay, this cell cycle, it includes the cell growth, regular things that a cell goes through in its reason for being that particular cell, and it's also going to include the division of that cell when that cell divides. There are 5 phases. Let's get the names of them first. You have the first growth phase, you have the synthesis phase, the second growth phase, you have the division of the nucleus, and you have the division of the cytoplasm. Those are the major parts of the cell cycle. Start looking at the details of each one. Interphase. Interphase is the largest part of the cell cycle. Okay, it's going to include the daily processes that a cell goes through, its growth, and all of that stuff. So if you look at this little picture down here, the green and blue and orange parts of the say this would be a cell. All right, very simplistic way of looking at it. But if this were a cell, this whole circle, this pie chart, then the green and blue and orange parts of the cell cycle would be interphase. Okay, and interphase includes the G1 phase, which is the first growth phase. This is just where the cell is going through its daily routine. Okay, it's growing, it's carrying out cell transport. If it's a chloroplast, it's going through photosynthesis. If it's a mitochondria, if it has mitochondria in it, it's carrying out respiration, obviously. It's transporting proteins, it's getting rid of waste, so it's doing all of the normal stuff that a cell would do in the first growth phase. That is the green section of this cycle picture. Then the S phase, the synthesis phase, is going to be the blue section, and this is where, okay, the cell knows that if it's going to make a new cell, it has to have the exact same copy of DNA to put into that new cell that it already has. The exact copy. It can't be missing a piece, it can't have extra pieces, it can't have something messed up in that DNA. So in the synthesis phase, it is going to copy its DNA exactly how it is to make sure there's an exact copy that it can put into the new cell. That's going to be the blue section, if you will, of the cell cycle picture here. And then the third part of interphase is going to be the second growth phase. Okay, this is where the cell is getting itself ready for cell division, to divide. Okay, it's prepping itself. It's moving things around. It's doing what it needs to do to make sure that everything is in the right place before it divides. This is the second growth phase. Okay, so you have the G1, the S, and the G2 phases in interphase. So if you look at this picture here, this red part of the circle is all interphase. Okay, and then within interphase, you have the G1 phase, you have the S phase, and then you have the G2 phase. Most of the cell, roughly 90% of the cell cycle is made up of interphase. All right, once interphase is done, then you get to mitosis. This is the division of the nucleus. All right, in this mitosis stage, what's going to happen is you're going to divide the nucleus. Okay, once the DNA has been copied and everything has been situated, you're going to divide the nucleus into 2 separate nuclei. Each is going to contain the complete set of the DNA. Okay, because remember you made a copy. It's going to contain a complete set of the chromosomes, which house the DNA. What's going to happen then are the chromosomes. The chromosome is made up of different parts, okay? These leg pieces are called chromatids, all right? And then the little piece in the middle that holds the two chromatids together is called a centromere. Remember, there are centrioles. And we've talked about those before, that will form within the cell, and they're going to be used to help separate these chromosomes into the right nuclei. Okay, so let's get into this. Let's get into the stages of mitosis and how this works. Okay. The first stage of mitosis is called prophase, and during prophase, the chromatin, remember the nucleus is full of chromatin, it's going to condense and coil around proteins, and it's going to make chromosomes. It's going to form these chromosomes. Chromosomes are made of chromatids, they're called sister chromatids, and they're going to be held together with centromeres. Spindle fibers will also form. The membrane of the nucleus will disappear. Okay, and then remember, everything is going, there's going to be doubles of everything, right? Because we copied this stuff. So this is happening in prophase. The chromosomes are appearing, are condensing, okay, and the spindles are forming, and then the nuclear membrane is disappearing. In metaphase, the second stage of mitosis, we're talking about the splitting of this nucleus, okay, in mitosis. What happens in metaphase is all the chromosomes, both sets of DNA that were formed through the interphase, all the chromosomes are going to line up along the middle of the cell. The spindle fibers that formed during prophase are going to attach to the centromeres that hold these chromatids together. Okay, so that's metaphase. And then in anaphase, which is the third stage of mitosis, the spindle fibers are going to pull those chromosomes apart. Okay, notice that here is a sister chromatid and here is a sister chromatid. This used to be one chromosome. Okay, but in anaphase, it's actually splitting them apart, and the spindle fibers are pulling them into opposite directions of the cell. And then the fourth stage of mitosis, telophase or telophase, what's going to happen is a new nuclear membrane or envelope is going to form around the two different sections of chromosomes that were pulled in opposite directions. Okay, and then you also get two new cells forming as a result. So you get these nuclear envelopes that are surrounding the chromosomes and the opposite sides of the cell. We'll call these daughter cells because they're going to be exact. They're going to be exact copies of each other. You'll start noticing that these daughter cells are going to be forming, and then there will be some type of, and we'll get to this later, but there will be a condensing of a wall separating here in the middle. So you'll see the two different parts forming in telophase. And then your last stage of the cell cycle is going to be cytokinesis. This is going to follow telophase. And what happens in cytokinesis is the cytoplasm is going to divide. That's what cytokinesis is. It's different in plants and animals. Okay. For animals, something called a cleavage furrow is going to form to help separate the two cells. Okay. For plants, the cell wall is going to be in the way, so the cell wall is going to kind of pinch itself in half and form something called a cell plate to help divide the two cells. And then here are some pictures. You have this cleavage furrow forming. You can kind of see that. You can see this through the electron microscope of the cleavage furrow forming. For the plant cell, you can see it for the cell plate forming there with the cell wall. But the same thing is happening in both things.